Today we will discuss about the hypersensitivity. So, as the name suggests, hypersensitivity means more sensitivity toward any kind of the antigen. Okay, so hypersensitivity can be defined as a state of altered immune response, or we can say like the increased immune response against an antigen, characterized by the hyperreactivity leading to the immunopathology. Okay, so suppose and this hypersensitivity reaction require a pre-sensitized state of the host. What it means? It means like in normal condition, suppose you encountered with certain kind of antigen or certain kind of the allergen, so some memory will be created inside our body, and once when next time this particular antigen or allergen encounter with your immune system, that time this memory cell will work as a immune defense mechanism. Okay, but in a very hyper reactive manner, and this hypersensitive reaction is like it is a localized kind of the reaction. Suppose you cause in means in your skin is the first entering side of your antigen, then only in this skin this hypersensitivity reaction will occur, not from the whole body. Okay, so this hypersensitivity reaction is a kind of local response. It is not the like throughout your body response. Okay, so hypersensitivity require three things. First thing is the allergen should be like pre-sensitized, or you should have already encountered with particular type of antigen or allergen, and once when you encountered, then second time when this antigen and allergen you get in, then this reaction will so its response in a localized site. Okay, and in a very hyper reactive manner, which can cause the immunopathology. Okay, and there are two type of the hypersensitivity. One is the immediate hypersensitivity, another one is the delayed type hypersensitivity. Okay, and this immediate hypersensitivity is mainly caused by the humoral immunity, or we can say like it is based on the antibody mediated immune response. Okay, and this delayed type hypersensitivity is majorly because of the cell mediated immunity, or T cell cytotoxic T cell mediated immune response, or cytokine based immune response. Okay, so this is the hypersensitivity. So there are there are means four different type of the hypersensitivity. First one is the type 1 hypersensitivity. Okay, so it is also known as the immediate or anaphylactic hypersensitivity. Okay, and it can cause by the IgE antibody. Okay, that you have to remember, like type 1 hypersensitivity is majorly preceded by the IgE antibody. Okay, so how it happened? The reaction occur on exposure to the allergen for the second time. Not means if you are encountering the allergen with first time, then this hypersensitivity will not work. Okay, normal your immune system will take care of this. But when you are encountering with the allergen for the second time, then this type 1 hypersensitivity will work, and the first exposure that is called as the sensitizing dose. Okay, because that time you are creating the memory for that allergen or antigen. Okay, so that is called the sensitizing dose, result in sensitization of host cell to the allergen. Okay, you will create the memory for the means for that particular allergen inside your body, and subsequent exposure will cause the reaction. Okay, so this is the mechanism. Like for causing the type 1 hypersensitivity, you have to firstly encounter with this antigen or allergen, then after that when you encounter with the second time, then this IgE mediated immune response will be stored in the particular location where you got the infection. That particular location only this response will happen. Okay, so it is it is also anaphylactic hypersensitivity called, because anaphylactic means it is like more extensive, more rapid more quickly it is happening. Okay, so like anaphylactic shock occur in the sensitized animal within the second to minute. Okay, so 15 to 30 minutes after exposure to the allergen, it is show the response. Okay, and after exposure to the antigen now called as an allergen, and sometimes the reaction may have a delayed onset, but it is not considered under the delayed type of hypersensitivity. It is the immediate type of hypersensitivity. Okay, but most of the time the reaction cause within 15 to 30 minutes. Okay, and the type 1 hypersensitivity reaction, the allergen are proteins basically with a molecular weight which is ranging from the 10 to 40 kilodaltons. Okay, so these are basically proteins, and there are different some pollen grain also can be cause of the type 1 hypersensitivity. It is nothing but means in simple term, if I will say, then it is a kind of allergic reaction. Okay, so suppose sometimes some bee will bite you, that time some inflammation happen inside our body, so that is also a kind of the type 1 hypersensitivity. Okay, so like that someone is having allergy towards the pollen grain, like that, so it is a kind of also allergy. And apart from that, if certain kind of the protein go inside, and within 15 to 30 minutes, if your body is showing response in a particular location, then it is called as a anaphylactic hypersensitivity. Okay, so for diagnostic test for immediate hypersensitivity include the skin test resulting in the will and flare reaction. Okay, and measurement of the total IgE and specific IgE antibody against the suspected allergen by ELISA. You can find out like how severe your inflammation is. Okay, like if it is showing more IgE antibody, then you can find out like you are having the severe hypersensitivity. Okay, through ELISA and through radio allergosorbent test. Okay, so most common is the ELISA only. Okay, so we means from patient we take the blood, and we check the antibody titer, how much antibody IgE antibody is present inside of the patient body. So through that we can identify like how much severe the hypersensitivity is. Okay, so it is the diagnostic test. Okay, so what is the mechanism of type 1 hypersensitivity? So the mechanism of reaction involve preferentially production of the IgE. Okay, so that you have to remember, like in case of type 1 hypersensitivity, the major antibody is the IgE antibody. Okay, and it will produced against the certain type of antigen, and in most of the cases it is the allergen. Okay, and in some cases only some kind of protein which is having the molecular weight 10 to 40 kilodalton, then that can cause the type 1 hypersensitivity, but most of the time it is called as the allergens. Okay, and IgE antibody is the major antibody which lead this type 1 hypersensitivity. Okay, and this individual which is prone to type 1 hypersensitivity preferentially produce the interleukin 4 and interleukin 13 that favors the IgE class switch. Okay, so these interleukins will enhance the IgE production as well as the IgE subtype. It will help to class switch. Okay, then it is having very high affinity for its receptor, that is the CD23, which is expressed on the surface of mast cell and basophil cell. Okay, and the Fc portion of IgE bind to this receptor. Okay, so yeah, here if we will see, suppose this is the type 1 hypersensitivity reaction. Okay, so firstly allergen, first time when it go inside your body, that time normal immune response will happen. Okay, so MSC will presented this allergen to the CD4, and CD4 again send signal to the B cell to produce for this memory cell as well as the plasma cell. Okay, so this plasma cell, what it will do is, when second time this allergen again come, then this plasma cell will produce the IgE antibody, and this IgE antibody is having the Fc receptor. We have seen that in antibody structure there is two part. One is the Fab domain, which is present here, which bind to the antigen, okay, and another one is the Fc domain, okay. So this Fc domain is having receptor inside the mast cell and the basophil cell, okay. So this black color is the receptor for the IgE antibody Fc receptor, okay. So when this mast cell Fc receptor bind to the IgE antibody, then what will happen is this mast cell or either basophil cell will be activated, okay, and it will start producing some granular part, that is most of the time is the histamine, okay. So it will cause the degranulation, okay. So yeah, here it is saying the degranulation, mast cell degranulation is proceeded by the increased calcium ion influx, which is a crucial process, okay. So through the calcium ion channel influx, what will happen is degranulation inside of the mast cell will happen. So in degranulation there is lots of histamine is present, okay. So histamine, what it will do is it will a kind of anti-inflammatory molecule, okay. So it will produce more, so it can, so that it can neutralize this allergen. So that's why degranulation will happen, and the side effect of this degranulation, what will happen is smooth muscle cell, then small blood vessel will be also small, and it also affect the mucus gland, and also affect the blood platelet, and affect the sensory nerve ending, okay, and also stimulate the eosinophil so that it can quickly handle out this allergen, okay. So this degranulation of cells result in the synthesis and secretion of inflammatory mediators such as the platelet activating factor, leukotrienes, bradykinin, prostaglandin, and cytokine that contribute to the inflammation, okay. And the reaction is amplified, means increased by the platelet activation factor, okay. So once this degranulation will happen, that time certain kind of molecule will be produced, and one of those molecule is the platelet activation factor, which will increase this reaction. So that's why it is called as the hyper response, right, because this molecule will enhance those particular immune response. So that's why the the allergen can quickly neutralize by the immune system, okay. And because of this there is a release of the histamine, heparin, and vasoactive amines, eosinophil, chemotactic factor of anaphylaxis, and neutrophils. All this type of molecule will be secreted during this response, okay. And this agent cause the early phase of the allergic reaction. As I told you, like it will happen within the 15 to 30 minutes after the allergen get enters your body, okay. And the last phase allergic reaction may begin several hours after the exposure to antigen. The cell bound IgE on the surface of basophil of sensitive individual bind a substance called the histamine releasing factor by the macrophage and lymphocyte, causing further histamine release, okay. So this whole process is carried out by three major things. First one is the IgE antibody, okay, which will produce and through the plasma cell, and it will go and bind to the mast cell where it is having the receptor for it, okay, either mast cell or either the basophils. Then this mast cell and basophil will be stimulated, and it will start producing the degranule particles, okay. And these degranule particles are the prostaglandin, bradykinin, cytokines, all this type of particle it will produce. And one of the major particle is the histamine, okay. It will increase the response, okay. And for quickly neutralizing this allergenic reaction, and it is a kind of immediate immune response, okay. So this is the type 1 hypersensitivity. Then the pathology of type 1 hypersensitivity. The primary cellular component in type 1 is the mast cell or basophil, and the reaction is amplified by the platelet neutrophils and eosinophils, okay. and the reaction may involve either skin, it can occur either skin, eyes, and nasopharynx, and gastrointestinal tract, and like the reaction may cause from the minor inconvenience to the death, okay, but it is more likely like a inflammatory reaction, so death is not so common in this case, okay. In most domestic species the lungs are the primary target organ, and like dog and all, okay. And the IgE mediated disease in human include the systematic anaphylactic shock, asthma, and hay fever. These are the like example for the type 1 hypersensitivity, okay. Asthma is one of the most common type 1 hypersensitivity, okay, where the like because of some kind of allergen you are continuously you are having problem to taking breath, so that time what is happening is more production of the IgE, and so that's why it is causing the hyperimmune response, okay. And conjunctivitis, skin reaction, food allergy is also kind of type 1 hypersensitivity, okay. And the IgE mediated disease in animals include systematic anaphylactic shock. Yeah, all these are the different types of example of type 1 hypersensitivity. Some of example you can like remember for exam point of view, like the asthma, hay fever, and this conjunctivitis, and yeah, atopic dermatitis. These are the like and the most common is the anaphylactic shock, okay. So this thing you have to remember, like these are the example of type 1 hypersensitivity. Then we are having the second type of hypersensitivity, okay, that is the type 2 hypersensitivity, which is also antibody mediated cytotoxic reaction, okay. And reaction occurring when an antibody bind to antigens located on the surface of the cell, okay. And this is also a kind of immediate type of hypersensitivity, okay. And it is mediated primarily by the antibodies of IgM or IgG class, okay. So
Excuse me,
they are,
ma'am.
yeah,
Ma'am, have you enabled recording?
yes, it is enabled.
Okay, okay, okay. Since in my laptop it is not showing, that's why I just asked.
No, 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 it is recording.
Okay, okay. Thank you, ma'am. Proceed.
Yeah. So this is the type 2 hypersensitivity, and it is basically caused by the IgM or IgG class of the antibody, not the IgE antibody, okay. So when it is happen when your antibody bind to the antigen, and which is located on the surface of the cell, okay. So mostly suppose this is the RBC, and if your RBC is having antibody on the surface of the cell, then when it encounter the antigen, then it is called as the like type 2 hypersensitivity, okay. And this type 2 hypersensitivity also require like a pre-exposure of the antigen, okay. All kind of hypersensitive sensitivity require the pre-exposure of the antigen or allergen, okay. But the processing method is different in all the cases. Like in case of type 1 there is IgE play role, and mast cell and basophil play role. Here the IgM and IgG antibodies play role with the help of RBC, okay. And this bound antibody can cause the cell lysis by activating the either activating the complement pathway, or through the antibody dependent cell cytotoxicity, okay. So there are two type of type 2 hypersensitivity for neutralization of the antigen. One is the complement pathway, okay. So there are a different complement factor is there, so it will activate a cascade of the complement system to remove those antigen, or it can like activate the antibody dependent cell cytotoxicity, okay. So this antibody will go and stimulate certain kind of the cells, and that cell will produce certain kind of the cytokine to neutralize these antibodies, okay. So that is called the antibody dependent cell cytotoxicity, okay. and many different antigen may trigger this cell destruction, but an infection in a genetically predisposed animal appear to be a major triggering pathway. Okay, and uh, yeah. The example of this type 2 hypersensitivity is most common is the hemolytic anemia, blood transfusion, okay, and erythroblastosis fetalis. Okay, these are the most common example of the type 2 hypersensitivity. Okay, and IgM or IgG antibodies are made against self antigen either as a result of the failure of the immune tolerance or cross reactive antibody can develop during the infection. Okay, like in the example, from the example we can see like blood transfusion, if certain mismatch happen, then this antibody can be that much hyper, like it can be cross linked with your antigen also, the self antigen. Okay, so it can cross react with your like self antigen, so which may bind to the normal tissue antigen and trigger the antibody mediated cytotoxicity. Okay, and this subsequent binding of these antibodies to the surface of host cell lead to type of process, either it can lead to the opsonization and another one is the opsonization or complement system. Okay, and another one is the antibody dependent cell cytotoxicity. Okay, so in what happen in case of opsonization is nothing but it is the phagocytosis process, but it is the increased phagocytosis process. Okay, in like in case of opsonization, you are targeting this, suppose this is your antigen, so when antibody bind to this antigen or antigen, then what is happen, you are marking this antigen to your immune system, like it will act as a like marker or the identification mark, like that, so that your immune system can recognize this particular antigen and do the phagocytosis. But in normal case, phagocytosis is normally happen for the like removing the death means debris or some death cell too from our body. Okay, but this process through the opsonization we are like enhancing this process, so that enhancement is called as the opsonization, and this antibody which is tagging to this antigen is called as the opsonin. Okay, and it will lead to the complement pathway. So there are different factors of the complement pathway which will lead to the cell death or apoptosis of this antigen. Okay, and second thing is the antibody dependent cell cytotoxicity mediated destruction of the host cell, where NK cell attached to the Fc portion of the antibodies. Okay, there like in case of type 1 hypersensitivity, mast cell is having the Fc receptor. Here in case of the ADCC, there is NK cell is having the receptor for the Fc region. Okay, so it will lead to the not degranulation, it will lead to the secretion of certain kind of cytokine so that it can neutralize the antigen. So this is the opsonization process. Okay, so after means when antibody attached to the cell, then once the it encounter to the antigen, it will lead to the complement activation. Okay, and this complement activation stimulate the some phagocytic molecule like the macrophage and dendritic cell, so that this antigen can be engulfed by this particular cell and processed by these cells. Okay, and this process is enhanced, that's why it is called as the hyper sensitivity. Okay, and complement after this phagocytosis, what will happen is it will represented through the complement system, and so that this complement factor will take care of this to neutralize this antigen. This complement system will be like activated and do the neutralization of this antigen. Okay, So the pathology of the type 2 hypersensitivity is it is mediated by the antibody of IgG and IgM toward the antigen which is present on the cell surface or the extracellular matrix. Okay, and the most common cells involved are the blood cells. Okay, and apart from that there is the NK cell or natural killer cells. Okay, and the outcome may be hemolytic anemia if RBC are involved, leukopenia if WBC are involved, or if thermocytopenia if platelets are involved. Okay, under some circumstances a cytotoxic attack on vascular epithelial cell will cause a vascularity with local vascular leakage. Okay, because this is the hyper means a local type of reaction, so it will not affect your whole body, it will only affect a certain region of the body. Okay, so yeah, it will lead to the complement system or neutrophils or dendritic cell macrophages like that. Okay, and which cells generally do the phagocytosis. Okay, so that type of cell it will stimulate, or it can through stimulation it can also stimulate the those cells which produce the cytokine to neutralize this. Okay, so one of the most common example of type 2 hypersensitivity is the erythroblastosis fetalis. Okay, so in that case what will happen is in the first pregnancy if mother is Rh negative, so they in mother's body there is no Rh antigen. Okay, but this baby is having the Rh positive antigen. Okay, Rh positive antigen. So through placenta what will happen is during the delivery some Rh specific antigen will cross the placenta and come to the inside of the mother's body. Okay, and it will produce because before that mother is not having this Rh antigen. Okay, so first time this Rh antigen is coming inside the mother's body. So as a foreign particle what mother's body will do is it will create some certain kind of the memory cell for this particular Rh antigen. Okay, when the mother is second time got pregnant, that time this Rh antigen of means this baby's Rh antigen will cause as a second allergen for the mother. Okay, so that time when it cross the placenta of mother, then it stimulate this memory cell which is already present inside the mother's body. It through the placenta, this memory cell will go to the baby's body, and it will cause to the it will produce the IgG antibody, and which can cause the erythroblastosis fetalis, which can lead to the abortion of the second child. Okay, so this is a type 2 hypersensitivity. Okay, and apart from that there is some autoimmune disorder is also coming. Another second one is the uh, Graves disease. Okay, so in that Grave disease what is happening is antibodies are made against the thyroid stimulating hormone receptor of the thyroid cell, leading to the faulty thyroid function. Okay, so here you can see there this is the thyroid epithelial cell, and here it is having the thyroid stimulating hormone receptor. Okay, so antibody will come and here bind to the this receptor. Okay, this thyroid stimulating receptor, so it will lead to the faulty thyroid hormone function. Okay, yeah, because this receptor is specific for the thyroid hormone, not for the antibody against the TSH receptor, right? So when another molecule will bind here, so it will lead to the faulty function. Like that, here another syndrome is the multiple sclerosis. Okay, where the antibodies are made against the glial cells that make the myelin. Okay, so this protein that forms the myelin sheath that insulate the nerve fiber of the neuron in the brain and spinal cord. Okay, so here if you will see there is the production of the acetylcholine, and acetylcholine will grow and will go and bind to the receptor which is present inside the muscle cell, surface of the muscle cell. Then it will produce the myelin sheath which is essential for the protection of your neuron. Okay, so when antibody will go and bind to this acetylcholine receptor, then this acetylcholine will won't be able to bind with this receptor, so that the myelin sheath will not produce. Okay, so this lead to the multiple sclerosis. Okay, so in that case you will be feeling the fatigue, okay, or tiredness or muscle pain, like that symptoms you can find out. Okay, and yeah, these are the some drugs we will use for the hypersensitivity, like the penicillin, which is more common antibiotic. Okay, yeah, then type 3 hypersensitivity. Okay, so in type 3 hypersensitivity, the soluble immune complex are the formed in blood and are deposited in various tissue. Okay, and which will activate the classical complement pathway and cause the inflammatory damage. So in case of type 2, there is when the antigen encounter with the circulatory cell, then it can cause the type 2 hypersensitivity. But here in case of type 3 hypersensitive, what is happening is some of the complex is accumulated in a particular place. Okay, so that accumulation lead to the type 3 hypersensitivity. Okay, which will also lead to the complement pathway to neutralize like the antigen. Okay, and it is mediated by the soluble immune complex. They are mostly the IgG class, and although means IgM is not so much common, but it may be also involved. But in case of the type 2 hypersensitivity, there is both the antibodies equally involved, but here the IgG is most common. Okay, and the reaction takes here also, it is a immediate kind of the hypersensitivity, so it happen in 3 to 10 hours. Okay, and the antigen may be exogenous, like the chronic bacterial, viral or parasitic infection. Okay, which will go and accumulated inside our body, so that it can lead to the type 3 hypersensitivity. Okay, and the most common example of type 3 hypersensitivity is the systematic lupus erythematosus. Okay, and SLE, which is the short form of this name. Okay, and the antigen is soluble and not attached to the organ involved. Okay, and the prerequisite for the development of immune complex disease is the persistent presence of soluble antigen and antibodies. Okay, so means for happening this type of type 3 hypersensitivity, there is pre-existence of the soluble antigen as well as the antibody should be there. Okay, otherwise it is kind of type 1 or type 2 type of hypersensitivity. When it is already present and accumulated in a place, then it is called as the type 3 hypersensitivity. Okay, but it also lead to the through complement pathway, it increase the immune response. Okay, so the mechanism of type 3 hypersensitivity is soluble antigen antibody required, that is the IgG or IgM, mostly the IgG complex. Okay, and complex are normally removed by the macrophages in the spleen and liver. Okay, on the formation of excessive amount or large immune complex, this get means lodged in capillaries, then pass between the endothelial cells of blood cell, is specifically in the skin. Okay, and become trapped on the surrounding basement membrane beneath these cells. Okay, it is accumulated means under the skin or joint or the kidney. Okay, so this local region cause the type 3 hypersensitivity because of the accumulation of the large immune complex. Okay, and the antigen antibody complex then activated the classical complement pathway, the damage being caused by the platelets and neutrophils. Okay, and here the massive inflammation happened due to the complement protein C5a. Okay, so as you know, in complement system there are different type of the complement factors. So the major complement factor which cause the type 3 hypersensitivity is the C5a. Okay, and in that case, influx of neutrophils due to complement protein C5a, resulting in the neutrophil discharge their lysosome and causing the tissue destruction and further inflammation. Okay, so this complement C5a, what it will do is it will stimulate the neutrophils. Okay, so when neutrophil will be increased, then the lysozyme content will be also increased, which can cause the tissue destruction and further inflammation. Okay, so because of the hyper reaction, what it is doing is with the neutralizing antigen, it is also neutralizing the self organ or self tissue. Okay, so that's why most of the hypersensitive reaction is the lead to the autoimmune disorder, because they are that much hyper, like it is it should be in a maintained manner, so that it won't hamper our body's tissue or organ, right? But hypersensitivity lead to the autoimmune disorder because it is that much hyper, like um, it can affect our self antigen also. Okay, so yeah, then this aggregation of platelet resulting in more inflammation and the formation of the microthrombi that block capillaries. Okay, so once this inflammation will be high, then what will happen is capillary of the body will be blocked. Okay, so it can cause different type of the symptoms. Okay, because of the blockage. So the pathology of the type 3 hypersensitivity is the affinity of antibody and size of the immune complex are important in production of the disease and determining the tissue involved. Okay, so it should be very large, then only it can called as a type 3 hypersensitivity, and where it is accumulating, that also matters. Okay, and the lesion content primarily the accumulation contain majorly neutrophils and deposit of the immune complex and complements. Then macrophages infiltrating in later stage may be involved in the healing process. Okay, so some of the macrophage also involved in the healing process of that particular hypersensitivity. Okay, and the clinical signs are therefore variable but may include the fever, cutaneous sign, and ataxia, behavior change, non-specific signs such as the vomiting, diarrhea, and abdominal pain. Okay, so these are the symptoms of the type 3 hypersensitivity. So the example of the, sorry, here wrongly written. Yeah, it is the, yeah, these are the type 3 hypersensitivity. Okay, here the example is wrongly written. Okay, of type 2 hypersensitivity. Okay, so then we are having the type 4 hypersensitivity. Okay, it is the only one type of hypersensitivity which is showing the delayed type of hypersensitivity reaction. Okay, it takes more than 12 hours to show the symptoms. Okay, and which persist for the time between 48 to 72 hours. Okay, and it is mediated by the mostly cells, the cytotoxic T cells, and cause the inflammatory reaction to either exogenous or auto antigens. Okay, means it can cause for the foreign particle as well as the for the auto antigens also, but it is unlike the other three type of hypersensitivity where the antibody play major role, here the cytotoxic T cell play major role. Okay, the major cells involved are the T lymphocyte and monocyte and macrophages. Okay, and this reaction to exogenous antigens involve T cell and also antigen presenting cells all produces the cytokines. Okay, and that stimulate a local inflammatory response in a sensitized individuals. Okay, and uh, delayed type of hypersensitivity can't be transformed from an animal to another by means of antibody or serum. However, it can be transferred by T cells, particularly CD4 and Th1 cells. Okay, so it is because like other two types is mainly responsible by the antibody, but here it is majorly caused by the CD4 cell or T helper cell. So that's why it will, what it will do is it will go and secrete certain kind of the cytokine which will lead to the stimulation of cytotoxic T cell. Okay, and which will produce again cytokines to neutralize or to show the defense mechanism against this particular antigen. Okay, So this is the mechanism. So suppose this is the antigen, okay, which will go and bind to the dendritic cell, and this dendritic cell will represent to the T helper cell, okay. This T helper cell will produce certain kind of the cytokine, okay, and this cytokine will go and stimulate other type of the immune cell like macrophages, okay. And this macrophages, what it will do is it will cause the inflammation through producing these pro-inflammatory cytokines like interleukin 2, TNF alpha, and interferon gamma, okay. So CD8 T cell and CD4 T cell recognize this antigen in a complex with either type 1 or type 2 MSC antigen, okay. And this antigen presenting cell in this case are macrophages, and they release the interleukin 1, which further stimulate the proliferation of CD4 cells. These CD4 cells will release the interleukin 2 or interleukin interferon gamma, which is a kind of the pro-inflammatory cytokines, which together regulate the immune reaction. Other lymphokines involved in delayed type hypersensitivity reaction include the monocyte chemotactic factor or tumor necrosis factor alpha, also a kind of type 4 hypersensitive factor, okay. And this cytokine produced by the keratinocytes, APC, and T cell, and activate the CD8 cell, which destroy the target cell on the contact, while activating the macrophages produce hydrolytic enzymes and transform into multinucleated giant cells, okay. So this whole thing is nothing but here the antigen will come and bind to the this dendritic cell, okay. This dendritic cell like either it is having the MSC1 or MSC2 complex, it will represent to the T cell, okay. Majorly it is CD8 T cell, okay, which will produce certain kind of the cytokine. This cytokine will be like interleukin 2, tumor necrosis factor alpha, and interferon gamma, okay. So this will all again produce the macrophages, okay, and it will perform the cellular infiltration or swelling on that particular location, okay. So this is the complete mechanism of type 4 hypersensitivity. Then we will see the pathology. So type 4 hypersensitivity is involved in the pathogenesis of many autoimmune disorders, okay, or the infectious disease like tuberculosis, leprosy, and histoplasmosis, toxoplasmosis, leishmaniasis, and granulomas due to infection and foreign antigens, okay. And there are three variant of delayed type hypersensitivity as listed below. One is the contact, another one is the tuberculin, third one is the granulomatous, okay. So all this is happen after like 12 hours it is showing the symptoms, okay. And the delayed type hypersensitivity lesion mainly contain the monocytes and a few T cells, okay. The means it is the cytotoxic T cell mediated, so it is the CD8 T cells, okay. And yeah, so this like in case of, if we will see the example, the tuberculosis case, how we the testing of the tuberculosis, when we do, what we will do is, under, suppose this is the skin, so of the human body, okay. So for checking the tuberculosis, what we generally do is some antigen of the mycobacterium tuberculosis we injected inside this skin, okay. Limited amount of the antigen which doesn't harm for your body, just to identify like this person is having the tuberculosis or not. So that time what will happen, if this person is having the tuberculosis, then this particular site will swell and appear as a red color, okay. So from there we can identify that this this person is having the tuberculosis, okay. So this particular type of inflammation what is happening here, that is comes under the type 4 hypersensitivity, okay, because it is happening after 2 days, that's why it is a kind of delayed type of hypersensitivity, okay. So yeah, so this is the three different type of the delayed type of hypersensitivity. One is the contact, another one is the tuberculin, and third one is the granuloma, okay. So eczema in some skin disease, if you have seen, that is comes under the contact type of the hypersensitivity. Okay, and another one is the tuberculin, what I told you, like inside the skin we generally give certain some amount of the tuberculosis antigen, so that it can we can identify like this person is having tuberculosis or not. Okay, and granuloma is like it is in that particular site, the skin will be hard. Okay, so that is the like it is happening in case of the tuberculosis also and leprosy also. So this is the delayed type of hypersensitivity. Okay, so this is the four different type of means it is the uh, difference between the type 1, type 2, type 3 and type 4. Okay, so type 1 is majorly because of the IgE mediated hypersensitivity. This type 2 is IgG mediated cytotoxic hypersensitivity. Okay, this type 3 is immune complex mediated hypersensitivity. Okay, both the cases IgM and IgM is also involved, but in case of type 3, um, uh, it the major means how we will identify like it is type 2 or type 3. Here it should be bind with the antigen. Okay, so then only we it should be bind with the antibody should be bind with the antigen, then it is called as a immune complex. Okay, so that type of complex when lead to the hypersensitivity, then it comes under the type 3 hypersensitivity. But in case of type 2, what is happening is the antibody means pre-exist in the surface of the cell, then it will encounter to the antigen. Okay, and in type 4 it is a completely cell mediated antibody cell mediated hypersensitivity. Here the antibody having no role. Okay, and here like antigen induces cross linking of IgE bound to the mast cell or the basophil with release of vasoactive mediators. Okay, and here antibody directed against the cell surface antigen mediated cell destruction via either complement system or the ADCC. Okay, antibody dependent cell cytotoxicity. Okay, and here the antigen antibody complex deposited in various tissue. Okay, it is accumulated in the various tissue which induce the complement activation and ensuring the inflammatory response mediated by massive infiltration of the neutrophils. Okay, so this complex when accumulated inside of certain organ, then it will lead to the stimulation of neutrophils. Okay, and this neutrophil increment will also increase the release of lysozyme, which will neutralize this particular complex. Okay, here you can see that this immune complex will lead to the complement system, and this complement system will then lead to the neutrophils. Okay, this neutrophil will release this lysozyme to neutralize this particular antigen antibody complex. Okay, then in case of type 4, it is sensitized by the Th1 cells or T helper cell, which will release certain kind of the cytokine that activate the macrophages or cytotoxic T cell, which mediate the direct cellular damage. Okay, so this is the like antigen when presented to the T helper cell. Okay, it will release certain cytokine that is the pro-inflammatory cytokine. Basically it can means stimulate either activated macrophages or the cytotoxic T cell. Okay, then these cells further lead to the cellular damage. Okay, so means with neutralization of those particular antigen, it will also lead to the cellular damage. Okay, then typical manifestation include the systematic anaphylaxis and localized anaphylaxis such as the hay fever, asthma and food allergies. Okay, and here it is during the blood transfusion reaction, erythroblastosis fetalis and autoimmune hemolytic anemia, we can see the type 2 hypersensitivity. Okay, and in case of Arthur's reaction, and serum sickness, rheumatoid arthritis. In that case we can see the type 3 hypersensitivity. Okay, so this example you have to remember, like which type of disease comes under, like which kind of hypersensitivity. Okay, and in type 4 the most common one is the tuberculosis. Okay, or contact dermatitis, graft rejection. That time we have seen the type 4 or delayed type of hypersensitivity. Okay, so in case of type 1, it is the another level of the comparison. So here antibody is IgE, here IgG IgM, here also IgG IgM, but here it is the immune complex which stimulate this IgG IgM. Okay, here the like it is in released form, it is not like in a complex form. Okay, it is accumulated, but it is in the circulation. Okay, and this type 4 is not having any kind of the antibody participation. Okay, this antigen will be exogenous in case of type 1. Type 2 will be means this antigen will go and bind to the antibody which is attached to the cell surface. Okay, then it type 3 there is the soluble antigen antibody complex, but it should be accumulated in the organ. Okay, in type 4 there is the tissue and organ, the antigen will be. Okay, and the response time is all three is having the immediate type of immune response, it is having the delayed type of immune response. Okay, and appearance is the whirl and flare. Here the lysis and necrosis you can see, here erythema and edema necrosis, here also erythema and induration you can see. Okay, and histology if we will see, the basophil and eosinophil is playing major role in case of the type 1 hypersensitivity. In case of type 2 there is antibody and complement system. Type 3 also complement system, as well as the neutrophils play major role. Okay, and here the T lymphocytes and monocytes or macrophages play role. Okay, and transferred with antibody. Here it is transferred with the T cells. Okay, and these are the examples for different type of the hypersensitivity reactions. The next question is, prior exposure of plant to pathogen is known to increase resistance to future pathogen attacks. This phenomena is known as systematic acquired resistance, hypersensitive response, innate immunity, or antibody mediated response. What will be the correct answer? So that's why the correct option will be systematic acquired resistance. And this antibody mediated response is also comes under the systematic acquired resistance. Then why not antibody mediated response? Because plant is not having antibody. Okay, so that's why the correct option of this question will be A, systematic acquired resistance. Understood? Everyone
Yes, ma'am.
understood why it is systematic acquired resistance? Because in the question it is asking about the complete plant, not about a particular location. If it is asking about a particular location, then that time hypersensitive response will be the correct answer. Okay, but here it is asking about the complete plant, so that's why like the answer will be option A. In under this acquired resistance, antibody mediated response is also coming, but if here it is the like animal based pathogen it is talking about, then you can say like antibody mediated response will be also a correct option. But here it is asking for the plant, so that's why in plant there is no antibody mediated response, so that's why option A will be the correct answer.